In order to understand how urine is made, we need to understand the nephron structure. The nephrons are considered the structural and functional unit of the kidney. Those are the building blocks of the kidney. And each kidney has about 1 million healthy nephrons. The nephron is made out of a renal corpuscle, and extending off of that corpuscle, there are small tubes known as tubules. The first step of making urine is known as filtration, and that occurs in these renal corpuscles. And then as the filtrate passes through these tubules, it is changed. So some things are important to the body and we want to keep them. So for example, glucose and amino acids, we don't want to lose that in urine. So that is a process known as tubular reabsorption. And other things are toxic and we want to actively secrete it into the filtrate, and that would be known as tubular secretion. So I kind of give out the 3 steps of urine formation. That is filtration that happens in the corpuscle, followed by tubular reabsorption and tubular secretion. So what does this renal corpuscle look like? It is made out of a tuft or a group of capillaries, and those capillaries are known as the glomerulus or the glomerular capillaries. These capillaries are surrounded by a very thin layer, just made out of actually one layer of epithelium that surrounds it as a capsule, and that capsule is known as the Bowman's capsule. So the renal corpuscle is the glomerulus surrounded by its capsule. And then from that capsule, the capsule extends, so that one layer of epithelial cells keeps on extending beyond the renal corpuscle and becomes the tubules. Now here you can see what the nephron looks like. So this right here is the renal corpuscle. You could see the tuft of capillaries or the glomerular capillaries on the inside, surrounded by Bowman's capsule. Extending off of that, you can find all of these tubules. They do have names and we want to be familiar with those names. So these mess of tubules that are very close to the corpuscle is known as the proximal tubule or the proximal convoluted tubules, abbreviated PCT. And then we have the nephron loop. So there is the descending limb and then followed by the ascending limb of the nephron loop. And then there is the distal convoluted tubule, abbreviated DCT, or the distal tubule for short. The capillaries that are inside of the corpuscle comes from this little arteriole known as the afferent arteriole. It breaks down to the capillaries and then exits as an efferent arteriole. So again, in the process of making urine, filtration happens here, what now becomes known as filtrate, and that filtrate will pass through these tubules and changed. So we talked about tubular reabsorption and tubular secretion. We'll get into the details of that in one of the next tutorials. Now here you can see the 2 different kinds of nephrons. So there are nephrons found in the renal cortex. Those are known as the cortical nephrons. These are make about 85% of the nephrons in the kidney and obviously are responsible for making urine. They are not involved in the what is known as the renin-angiotensinogen system. We'll get to that in a moment. You can also see that it's surrounded by a whole bunch of capillaries. These capillaries are known as peritubular capillaries. If you break down the different parts, so here again is the renal corpuscle, followed by the proximal convoluted tubule, then the descending limb, ascending limb of the nephron loop, and then the distal convoluted tubule, and then everything gets to be collected in this collecting duct. If you look at this nephron, This nephron is at the junction between the cortex of the kidney and the medulla of the kidney, and that's why they're known as the juxtamedullary nephron. Those make about 15% of the nephrons, and these nephrons are the ones that are responsible and maintain that renin-angiotensinogen mechanism. In addition to the peritubular capillaries, you see here that the nephron loop is related to these capillaries or these blood vessels found in the medulla, and these blood vessels are known as the vasa recta. Now for more details about the renal corpuscle, that the capillaries that are found in the glomerulus are fenestrated, meaning that it has windows, and those gaps are actually bigger in the glomerulus than in any other capillaries in the body. They allow fluid to pass through it by filtration. Pretty much almost everything can be filtered. There are exceptions. For example, the big plasma proteins cannot be filtered. Our blood cells, like RBCs and white blood cells, are not filtered either. So the bigger sized molecules are not allowed to be filtered out. And the Bowman's capsule are made out of cells known as podocytes. Podocytes because it looks like feet. They have like these finger-like projections, and in between these finger-like projections there are these little slits. Okay, so I'm going to go to the image to kind of explain what this looks like. So that right here, again, is the corpuscle. You can see the afferent arteriole bringing blood in. It breaks down into these glomerular capillaries, and then the blood exits through the afferent arteriole. Before we go into more detail about the structure of the renal corpuscle, I would like to get your attention to the fact that the afferent arteriole's diameter is wider than that of the efferent arteriole, meaning that more blood is coming in than what is leaving the glomerulus. And that maintains a high hydrostatic pressure inside of these capillaries that allows for filtration to occur. It also allows for the kidney to regulate filtration through changing the diameters of these arterioles. So we'll go through a couple of scenarios. If we need to increase the glomerular filtration rate, or the amount of filtration that is happening in the renal corpuscle, the kidney can do so by either dilating the afferent arteriole or constricting the efferent arteriole. In both of those scenarios, you are increasing the hydrostatic pressure in the glomerular capillaries. Well, what if you want to switch that scenario and the kidney needs to decrease glomerular filtration? It can do so by either constricting the afferent or dilating the efferent. So in both scenarios, again, that leads to the decrease in the hydrostatic pressure here, and that decreases the overall filtration. Now, going back to what the structure of the renal corpuscle is, these capillaries are covered by these podocytes. You can see here a nucleus, and you can see these foot-like projections with these finger-like projections coming out. And they're little tiny slits in between. Here again, this is the capillary with the gaps in between the endothelial cells of the capillaries. We know this is a capillary because there is a white blood cell in there. And then these are the finger-like projections with these very tiny slits in between. So for things to be filtered out, it would have to fit through this gap and through that gap. So what can be filtered is water, electrolytes, glucose, amino acids, fatty acids, vitamins, urea, uric acid, and creatinine. So some of these are waste products that we actually do want to get rid of, but you can see here things that are important to the body. So there's glucose, amino acids, for example. that the body needs, vitamins. Okay, we do not want to get rid of them and excrete them through urine. And that is why, as that filtrate goes off to the tubules, they will be reabsorbed back into the capillaries. Okay, you'll see here that the bigger cells, like bigger plasma proteins, the blood cells, and so on, are not going to be filtered out. Those are going to remain inside the glomerular capillaries. Now, Bowman's capsule is made out of a single layer of epithelial cells found on a basement membrane. For the capillaries that are associated with the nephrons, we kind of talked about these already. There are the glomerular capillaries inside of the, surrounded by Bowman's capsule. We talked about the afferent and the efferent arteriole and how we can change the filtration rate by changing the diameters of these arterioles. Okay, so that you want to be comfortable with those different scenarios. Now, the rest of the capillaries related to the nephron surrounding the tubules are known as the peritubular capillaries. The blood vessels that are related to the nephron loop in the juxtamedullary nephrons, those are known as the vasa recta. The two different kinds of nephrons, we talked about the cortical nephrons. Those are found in the renal cortex. Those make about 85% of our total nephrons. They have a very short, or sometimes they even do not have the loops of Henle, which is another name for the nephron loop, and they do not contribute to what is known as the hypertonic medullary interstitium, and we'll go over that in more detail when we get to that point. The second group of nephrons are known as the juxtamedullary nephrons. Those have very longer loops of Henle. They are found at the junction between the renal cortex and the renal medulla. They are responsible for generating an osmotic gradient in the renal medulla that is responsible for reabsorption of water. And again, I'm going to talk about that more in detail when we get to that part. It makes about 15% of all of our nephrons, and the nephron loops are close to those blood vessels in the medulla that we call the vasa recta. Now, the juxtaglomerular nephrons have an apparatus known as the juxtaglomerular apparatus, made out of different kinds of cells. There are two different kinds of cells there. There are the juxtaglomerular cells and the macula densa cells. The juxtaglomerular cells are stimulated when there is a decrease in stretch. So these juxtaglomerular cells are found in the afferent arteriole. If blood pressure goes down, these baroreceptors are going to feel that decrease or detect that decrease in blood pressure. Now these cells, these juxtaglomerular cells, are going to start secreting renin. Renin is an enzyme that has an effect on our blood pressure and sodium balance. So you might want to go to the endocrine system and in order to remember what were the effects of renin. The other group of cells are known as the macula densa cells, and these are very specialized cells that are found on the ascending limb of the loop as it turns to become the distal convoluted tubules. These macula densa cells sense changes in the osmolarity of the filtrate. So changes in the sodium chloride content of the filtrate. And in doing so, it can regulate how much sodium. Again, you want to always combine sodium with water. How much sodium and water is the body going to use? So it helps in regulation of sodium balance and blood pressure. You'll see that both of these cells have the same function, but in different ways. So the juxtaglomerular cells secrete renin, and these are stimulated by a decrease in stretch, meaning a decrease in blood pressure, while the macula densa cells detect changes in osmolarity of the filtrate. So they detect changes in the sodium chloride content of the filtrate. Again, both of these cells together make that apparatus known as the juxtaglomerular apparatus. Now we can see a picture of that apparatus right here. This right here is the corpuscle. You see the afferent arteriole entering, breaking up into the capillaries, and then the blood leaves as the efferent. These blue cells, those are the juxtaglomerular cells. Again, these cells detect changes in blood pressure in the afferent arteriole. The baroreceptors detect decrease in blood pressure, so there will be a decrease in firing from these receptors, decrease in stretch in those receptors, and that stimulates these juxtaglomerular cells to produce renin. Now, the macula densa cells are found at the very top of the ascending limb as it curves to become the distal tubules. These macula densa cells detect changes in the osmolarity of the filtrate, not blood, osmolarity of the filtrate. And according to that osmolarity, it will adjust how much sodium will be lost in urine. Now, the next tutorial will be talking about the whole process of urine formation.